I'm clocking in to give you a nice kind of continuum from our last lecture on mutations. So just to recap, mutations are any alteration in DNA sequence or the changing of a structure of a gene that results in a variant form. And this variant form will persist through cell division, become fixed in the genome, and voila, you have a mutation that can then be passed on to subsequent generations. So when we talk about mutations, all right, some of them are bad, some of them are good, right? Some amount of mutation is actually beneficial. You need a small amount of change in order to allow genetic diversity. However, again, some of that change is bad. Actually, most are bad in regards to mutations because they can result in a non-functional protein and or genetic disorder. So this is where your DNA repair systems come into play. DNA repair systems act as a balancing mechanism for genetic change, where it maintains balance between DNA repair and mutation rate. So if the balance is normal, there will be very little genetic change. For eukaryotes and E. coli, approximately one nucleotide in every 10 to the 10th power is altered and passed on to the next generation. So for those of you who have trouble with exponents and logs, 10 to the 10 is 10 followed by 10 zeros. So that means there's a high amount of sequence that is accurate, that does not have some sort of error or change in it. So we define DNA repair systems, because you're probably wondering what they are. These are just collections of processes by which the cell is able to identify and then repair damage to a sequence of DNA. Now, it's crucial for you to understand that there's a distinct difference between damage and mutation. Figaro, stop it. Sorry, my cat is playing with the water bottle. Thank you, Figaro. So DNA damages can be recognized by enzymes, and thus they can be correctly repaired. A mutation arises when that damage becomes fixed. Now, because it's not corrected during replication and now exists as a base change that's present on both strands of the DNA, it can't be recognized by an enzyme. So once that happens, that mutation can no longer be recognized by an enzyme and thus cannot be repaired. So a damage, you have damage first. If that damage becomes fixed, it's a mutation. So it kind of leads into the formation of a mutation, but maintaining the understanding that the two should be regarded as different things. Again, damage has the potential to be repaired. Mutation, once it's there and once it's fixed, it cannot be repaired. So what are these actual sources of damage per se? Well, you can have the loss of bases, right? That creates these abasic sites where you have these apyrimidine or apurines. You could have base modifications, replication errors, and base convergence. These can generate mismatch nucleotide pairs. You can have failures in normal DNA metabolism by topoisomerases and nuclease. So this can actually generate single-strand or double-strand breaks. You can also have photo damage by UV radiation or these chemical agents and reactive oxygen species, which will typically modify the bases. All of these can serve as fundamental sources of damage. So your DNA repair systems will come in and will serve to kind of fix these damages. Now, the actual repair is dependent on many factors. For example, it's dependent on the cell type, the age of the cell, and the extracellular environment. All of those are going to influence the function, the actual type of repair that's used, and the efficacy of that repair. So there's common aspects of DNA repair systems. There's tons of different repair systems. I've narrowed them down to a select few that I feel are the most important and most often brought up in passing genetic conversation. But of all of them, again, there's these commonalities to them. So one is that they all require, or most of them require, an undamaged strand to serve as a template during repair. And that will serve as the template for the repair of the complementary strand, which contains that point of damage. And this is to allow viable reproduction, recreating a strand or making a fix that is actually viable and meaningful. The second is that there's redundancy in repair systems. So one type of damage can be repaired by more than one system, right? And this is simply to act as a fail-safe system. So for some reason, this repair system could not be put into place. There's this backup one that can be utilized. It's not like all is lost and everything is end of the world, doom and gloom. And the third is that they all use a specific set of proteins. In fact, most of these proteins come from components of DNA replication. They'll actually function in DNA repair. So for instance, DNA polymerase 1 functions frequently in your DNA repair systems. So it makes the system extremely efficient. And they'll use the same proteins for multiple functions, and again, the same repair systems for multiple sources of damage. Come on, there we go. So this is just looking at the actual accumulation of DNA damage. So you want your damage any damage that occurs in the DNA to be repaired, and repaired as quickly as possible. You don't want this accumulation of damage to occur because it can have very deleterious impacts on cell functioning and actual viability of that DNA. So again, you have all these different sources of different types of damage. So you have these exogenous sources, right? These your radiation, chemicals, toxins, pollutants. And then you have your endogenous sources. So replication errors, hydrolysis, these are all internal errors. These are kind of your environmental sources. So when you have the accumulation of DNA damage, that can lead to different types of cancers. You can have actual defects of cellular functions, or you can have cell death or cell suicide, apoptosis, or this permanent almost dormancy of cell functioning. That's what senescence is. And both of these will lead to aging. And then you start getting into this vicious cycle. So you have the aging of the cell. That's going to decline the efficiency and accuracy of any DNA repair system, right? You kind of start hindering that. You're going to increase the accumulation of DNA damage. So you start getting this positive feedback loop where if you have this accumulation of DNA damage is just compounding the amount of damage because it's influencing or the accuracy and ability for DNA repair to occur. So 3 main steps for how DNA repair occurs. It's not difficult. So first, that damaged or incorrect DNA is detected. Once it's detected, that damaged or incorrect DNA has to be removed. And once it's removed, you have to replace it, right, with the correct DNA. So you can't just leave a big gap there. You have to fill it in with the correct nucleotide sequence. So there are 5 principal repair systems that I'm going to be talking about. DNA replication repairs, these are these guys over here. They are those that occur immediately during or after DNA replication. So these are proofreading repair and mismatch repair. The others over here are not happening or relative to DNA replication. So they occur at all other times besides replication. And repair, again, usually only occurs on one strand, and that's to use the other strand as the template for repair. And this is, again, to allow for the correction to produce a viable product. So for proofreading repair, again, this is occurring during replication. This is one of your during replication repair systems, and it's mediated or catalyzed by DNA polymerase. So the DNA polymerase will add, it comes along, right? You're doing your normal replication here, and something happens. Maybe you have slippage or something stalls the polymerase while it's replicating or synthesizing that new strand of DNA, and it places or adds an incorrect nucleotide to that new strand. So you have this incorrect base pairing, right? So you should be having cytosine join here or base pair with this guanine nucleotide. Instead, you have your thymine. All right, so that incorrect base pairing changes the width and alters the shape of the double helix. And that change in kind of the appearance, I guess you could say, of the double helix allows the polymerase to detect the mispaired bases. So polymerase will then come along and it uses its 3' to 5' exonuclease activity to remove that incorrect nucleotide, right? So here's your incorrect nucleotide or an incorrect base pair here. So remember, exonuclease, this is an enzyme that can break nucleotides. So when polymerase moves 3' to 5', it can have exonuclease properties. 5' to 3' is when it has synthetic properties. Let's say that 5 times fast. Or to somebody with a lisp. Oh, that's a nightmare. So it can, again, break DNA by moving 3' to 5', builds 3' to 5'. Sorry, 5' to 3'. So your polymerase comes here, is able to detect that mispaired bases and will cleave them or break them using its exonuclease activity. Once that happens, it will start moving in the correct direction, and you can have the continuation of synthesis. So then you have the thymine removed, and assumingly a cytosine will then be placed. So mismatch repair. This occurs immediately after DNA replication. So here is your new little double helix product, right? And you have this mismatch base pair here, right? You have a thymine that's base paired to guanine. So that mismatch causes a bulge. Here it's a bulge. It can also cause a narrowing in the double helix. This will distort its structure, allowing the mismatch to be detected in this newly synthesized strand of DNA. So a set of proteins will cut out a segment of that newly synthesized DNA that contains that mismatched nucleotide. It won't take out both sections. Again, it just takes out just the segment on that strand containing the error. Does that, again, because you want to use this strand here for the template. So Polymerase will use, again, its 3' to 5' exonuclease activities or abilities to kind of cut this segment out. And then that missing patch will be replaced by correct nucleotides. Those correct nucleotides, again, will be placed or set down by DNA polymerase, traveling in your correct 5' to 3' direction. I should say reading your strand that's serving as the template for correction in the 5' to 3' direction. So when that fragment is filled, ligase will come in. You remember ligase from DNA replication. This will come in and it seals the edges of the fragment in the DNA backbone. So that way, again, you get this continuous sequence. There's no gaps in the backbone. It's like everything was fine and dandy to begin with. So now we're moving into the types of DNA repair systems that occur not during replication or have are not associated with actual DNA replication, that event at that time. They occur at a different time. So the first one we'll talk about is this interesting technique called direct reversal. It's a very simple technique, you could say. It simply deals with the direct removal of a group. So an incorrect here, a methyl group is added, and then direct reversal simply takes that methyl group off. So it's, again, relatively simple, and there are some several mechanisms in which the damage is directly reversed. Most often it's by a single repair protein without, and the great thing about it is that you don't have this incision made to the DNA backbone. So the downfall is that it only mediates the reversal of a relatively small set of what you would call DNA lesions. So it can only work or function on small sections of damaged DNA. But that's okay, because even though it operates on small sets, its simplicity and relatively high degree of accuracy make this mechanism, this direct reversal mechanism, or mechanisms of direct reversal, I should say, rather attractive to the cell. It's efficient, it's quick, and again, very easy. So there are three major mechanisms, per se, of direct reversal. You have photolyase, alkyl transferase, and dioxygenase mediated repair processes. So the whole concept of direct reversal, again, it might be a more efficient way of dealing with damages, especially for species who might, or specific types of DNA damage that may occur more frequently. So going back to those three mechanisms, the first one I said was photolyase. So this deals with damage that occurs because of exposure to UV radiation. Well, all species are continuously exposed to UV radiation, so it makes sense that you have a direct reversal repair mechanism for that that factor. So only a few types of DNA damage are actually repaired with direct reversal. The large one being your photolyase. Here you have this creation of what are called pyrimidine dimers, and they are created or the result from exposure to ultraviolet light or radiation. You also have alkylated guanine residues. These are another source of potential error. They can be modified by the addition of methyl or ethyl groups at the O4 position of the purine ring. So you see that being used or that type of damage being treated oftentimes with direct reversal. And that's kind of what we have going on here with the addition of this methyl group. So you have regular guanine, some type of reaction to a harmful chemical in the environment causes the addition of a methyl group to the oxygen, right? So now you get this methylated guanine, and then that's detected and can be quickly repaired by just removing this methyl group. So again, you're not causing any incision in the DNA backbone, you're simply removing that troublesome molecule or group. So interestingly, the repair of pyrimidine dimers by reactivation, this is a common repair system that you see, or this is what you typically see in prokaryotic and eukaryotic cells. Interestingly, photoreactivation is not universal. So many species, including us little human beings, lack this mechanism of DNA repair. We actually use a different type, more specifically nucleotide excision repair, to deal with damage from UV radiation. I don't actually know the whole reasoning or evolutionary purpose of that. I don't know if it's actually even been discussed or have found. That would be interesting to seek out. Anyway, moving on. So next runner-up, again, these are all repair systems that come into play for damage that occurs at any time in the system. So the next one is base excision repair. So this involves removing or excising one base. The chemistry in the cell, either naturally or by some sort of environmental change, causes you to remove that appropriate amino acid and replace it with an inappropriate base. So deamination, right, that's a change in amino groups, converts here in this example, it'll convert cytosine base, that should be here, to uracil. And the uracil, again, causes this bulge in that strand of the double helix, which can then be detected and removed. So that leaves this little space here in the DNA backbone. It's like somebody's lost a little tooth. So DNA polymerase will fill that hole with the correct base, and then again, either side of that once gap will be sealed by ligase. So you have a continuum of your sugar phosphate backbone. So then there's nucleotide excision repair. So this was one that I mentioned just a couple of slides ago, talking about the direct reversal repair systems. So this is often caused by sun exposure, right? This is triggered by your UV radiation. So here, UV radiation causes thymines that are in the DNA to covalently bond at nitrogenous bases. And this produces something that's called a thymine dimer. So this makes, that's this weird looking thing here. So this makes the double helix very stiff at that site. You have an enzyme called photolyase, which is a protein that's activated by UV exposure, will specifically recognize this thymine dimer. Photolyase is another kind of repair system for that thymine dimer. So once the dimer has been detected by your photolyase, it's kind of like your alarm signals are now all going off. The surrounding DNA is opened up to form this bubble. Enzymes will then come in, they cut the damaged region out of the bubble, right? So here it's been cut on either side, so you have the incision to the DNA. It's cut, and removed, and then a DNA polymerase will replace that excised DNA with correct nucleotides. And then your ligase will come in and it will seal the gaps between that newly created segment with the proper base pair. Now, I apologize if you hear scurrying around in the background. That is, again, my cat Figaro. I gave him a new little toy to play with that he is quite enjoying. Usually has a squeaky dinosaur, but I don't know where that poor little squeaky dinosaur has gone. He was looking very upset and forlorn that he lost his favorite little squeaky toy, so I had to get him a new one. Poor life decision. Anyway, so all of the ones that I just discussed previously, they deal with these single-strand breaks, right? Or errors that occur on just one strand. These two that I'm going to be talking about, your non-homologous and homologous, they deal with what are called double-strand breaks. So some types of environmental factors, such as your high energy radiation, can cause these double-stranded breaks in the DNA. So here you're actually splitting a chromosome in two. Now this kind of DNA damage, this is like the type that is linked in these superhero origin stories and comic books, and with, for a real life example, the disasters in Chernobyl. So DNA stranded breaks are very dangerous because large segments of chromosomes and the hundreds of genes that they contain may be lost if those breaks are not repaired. Now, there are two steps, again, for those repair systems for your double-stranded DNA breaks. As I said, your non-homologous and your homologous recombination pathways. So in non-homologous end joining, the two broken ends of the chromosome are simply glued back together. This mechanism is pretty messy and typically involves the loss or sometimes addition of a few nucleotides at that cut site. So non-homologous end joining tends to actually produce a mutation, but this is better than the alternative, which is the loss of that entire chromosomal arm. So it can also lead to the creation or elimination of chromosomes, which can then lead to speciation. So that can either be a good thing or a bad thing. The other is homologous recombination. So this uses a homolog, hence the name. As such, this can only occur in eukaryotes, and that's because prokaryotes are haploid and therefore they don't have a homolog. So information from the homologous chromosome that matches the damaged one, in other words, you're using the sister chromatid, if the DNA has been copied, is used to repair that break. In this process, the two homologous chromosomes will come together, and the undamaged region of the homolog, or chromatid, is used as a template to replace the damaged region of the broken chromosome. So because you're using a homolog, something, again, that's viable, you're using a real template, your homologous recombination is much cleaner than non-homologous end joining, and it does not usually cause any mutations. From it, you're likely to create something that's actually viable for the cell. And again, that's all because you're using a homolog for an actual direct template of repair. Now, when you lack appropriate DNA repair systems, obviously that has consequences. I was talking earlier about the accumulation of DNA damage. Lacking DNA repair systems in and of itself means that you're going to have these mutations that can cause genetic disorders. So evidence for the importance of proofreading and repair mechanisms again, that comes from your human genetic disorders. In many cases, mutations in genes that encode your proofreading and repair proteins have some association with hereditary cancers. So these are cancers that will run in families. This is like the hereditary breast and ovarian cancers, which is HBOCs, and your hereditary non-polyposis, I think it's called, colorectal cancer. Simple terms, Lynch syndrome. Another great example is your xeroderma pigmentosum, right? So this is a rare recessive genetic disorder wherein people with this genetic disorder become extremely sensitive to UV light. And it's caused by mutations that are affecting the nucleotide excision repair pathway. When that pathway doesn't work, those thymine dimers and other forms of UV damage won't be repaired. So people with xeroderma pigmentosum develop severe sunburns from just a few minutes of exposure to the sun, and about half will get skin cancer by the age of 10 unless they avoid the sun. And again, it's all these skin abnormalities. Also have developmental and neurologic defects. But that's just gives you a really great example. Not great, it's horrible, but it gives you an idea of how deleterious it can be to the human body or any organism to lack appropriate repair systems. They are very important. So that is all I have on DNA repair. I hope that was interesting and did not put you to sleep. If it did, then hopefully I can get a Nobel Prize for finding a cure for insomnia. But other than that, I hope you have a good rest of your day.